Hello and welcome to some biology class. Friends, today we shall discuss about adherence junction. Adherence junctions are the cell junctions present in epithelial tissue. They are more basal than tight junction. You can also call them as junctional complexes. Adherence junction initiate contact between cells and mediate the maturation and the maintenance of the contact. They are protein complexes composed of different type of proteins. Various components of the adherence junction are E-cadherin. E-cadherin means epithelial cadherin. This protein is the main transmembrane component of the adherence junction. Second component is the actin microfilament of the cytoskeleton. And the third component include different type of proteins which link the E-cadherin to the actin cytoskeleton. These proteins are called as anchor proteins. Various proteins that are present inside the cell like alpha-catenin, beta-catenin, gamma-catenin, vinculin, alpha-actinin, all of these proteins together serve as anchor proteins. These anchor proteins link the actin cytoskeleton to the E-cadherin. This figure shows the various components of adherence junction. This is the E-cadherin. E-cadherin is the main transmembrane component of the adherence junction. E-cadherin has 5 extracellular cadherin repeat domains, and this is the intracellular domain which is present inside the cell, and this is the plasma membrane. Two molecules of E-cadherin form a E-cadherin dimer. So here this is a E-cadherin dimer and this is another E-cadherin dimer. These two E-cadherin dimers are present in the plasma membrane of interacting cells or adjacent cells. And here you can see that this E-cadherin dimer bind with this E-cadherin dimer. So there is trans-cadherin interaction. And this is the intracellular domain. The intracellular domain bind with different type of anchor proteins. Various anchor proteins that bind with the intracellular domain are P120 catenin, beta catenin, alpha catenin, and a number of proteins like vinculin, actinin, formin 1, and so many proteins together serve as anchor proteins. These anchor proteins link the intracellular domain with the actin cytoskeleton. This is about the various components of the adherence junction. And this is the intercellular space. The intercellular space varies from 25 to 40 nanometer. So the extracellular domains of E-cadherin are present in the intercellular space, and the extracellular domain of one cadherin dimer interact with the cadherin dimer that are present in the plasma membrane of the adjacent cells. The E-cadherin and different types of catenins that are associated with it together control the formation, maintenance and function of adherence junction. It is the transbinding of extracellular domains of E-cadherins of adjacent cells that help to link the cells together. Now let us discuss about the various components of adherence junction in detail. First component is E-cadherin. E-cadherin is a single-span transmembrane glycoprotein. It is a member of the classical cadherin superfamily. It is a calcium-dependent adhesion protein. E-cadherin has 5 extracellular cadherin repeat domains. These five extracellular cadherin repeat domains bind with calcium ion. The binding of calcium ion with these domains form the correct conformational organization so as to ensure binding of extracellular domain of E cadherin of adjacent cells. The cytoplasmic domain, this is the cytoplasmic domain of E cadherin, bind with various proteins that regulate endocytosis, recycling and degradation of E. coli. These proteins also regulate intracellular signaling, gene transcription and local control of actin cytoskeleton. Within the intracellular domain of E. cadherin, there is a just membrane domain. This is the just membrane domain which is 94 amino acid long. The just membrane domain bind with P120 catenin and the carboxyl terminus domain bind with beta catenin. Cadherin mediated cell-cell adhesion is highly dynamic, enabling the reorganization and dispersal of cells. For example, during the epidermal to mesenchymal transition in development and in carcinogenesis. E. cadherin is the core transmembrane component of the adherens junction. This protein is required for binding and localization of a number of important cytoplasmic proteins that link the cadherin complex to the actin cytoskeleton and several signaling pathways. The third component of adherens junction is anchor proteins. Anchor proteins include different type of catenins like alpha catenin, beta catenin, P120 catenin and other proteins like vinculin and alpha actinin. Alpha catenin, beta catenin and P120 catenin are the members of catenin family. Beta catenin and P120 catenin are the key regulator of E. cadherin. Both are armadillo repeat domain proteins. P120 catenin bind with the just membrane domain of the intracellular domain of E. cadherin. It is believed that binding of P120 catenin to the just membrane domain of E. cadherin block factors like E3 ubiquitin ligase and other proteins of the endocytic machinery that tag and target E. cadherin for destruction and internalization. Another catenin is beta catenin. Beta catenin bind with the carboxyl terminus domain of the intracellular domain of E. cadherin. Beta catenin also bind with alpha catenin. Together they modulate the interaction with the underlying actin cytoskeleton. Alpha catenin is an F-actin binding protein and an homologue of vinculin. This protein forms an essential link between cadherin, beta catenin and the underlying cytoskeleton. Another anchor protein is vinculin. Vinculin has no enzymatic activity. It regulates adhesion by directly binding to actin and stimulating actin polymerization and by recruiting actin remodeling enzymes. In the absence of vinculin, cell-cell and cell matrix adhesion is dramatically impaired. This indicates an essential role of vinculin in human physiology. So, in the adherens junction, E. cadherin bind with different type of catenins to form adherin catenin complex, and the adherin catenin complex is linked with alpha-actinin and vinculin. Alpha-actinin and vinculin, these are the two proteins that are responsible for linking cadherin-catenin complex to the underlying actin cytoskeleton. E-cadherin is an calcium-dependent adhesion protein, and the morphological and functional integrity of adherens junction is calcium-dependent. Functions of adherens junction. Adherens junctions play multiple roles, including initiation and stabilization of cell-cell adhesion, regulation of actin cytoskeleton, intracellular signaling, and transcriptional regulation. Friends, this is about adherens junction. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you very much.